Bit of stuff to talk about still on the urea cycle, and then we will get into the last topic before the midterm, which is fat metabolism. So we'll be talking about the breakdown of fats today, and then the synthesis of fats on Friday, and then Monday will be the review session, and there's a quiz due on Monday as well, and then Wednesday is the midterm.
Should we try and take the quiz after Friday's lecture?
The quiz will include Friday's lecture. So I would take the quiz sometime over the weekend or Friday afternoon or whenever you want before Monday. Monday at 11:29 a.m. is when it will turn off. Okay, so we talked about nitrogen metabolism a little bit last time, and so we're basically looking at the process by which dietary protein or amino acids from other parts of the body converge into the liver, and then the nitrogen gets excreted. But in the process of that, there's a sort of a complex connection between the nitrogen excretion process and the TCA cycle, and there's this extra, what's called the aspartate argininosuccinate shunt. Nice, easy name for us to remember. It basically is a way in which the urea cycle and the citric acid cycle are connected. It shouldn't be drawn as large as this because it's not nearly as big of a cycle or anything like that. It's just a couple of reactions on the side, really. Amino acids come in. They don't usually get used for energy purposes. They can if there's a massive excess of protein in the diet, or especially if there's a starvation condition, at which point the cell will start to sort of eat itself up, so to speak, in order to provide enough ATP to keep itself going. We're not considering catabolic pathways in any detail, at least in terms of the carbon skeleton. Right? So the carbon skeleton here, protein comes in various ways, and the carbon skeleton of the amino acids in one way or another, and sometimes it takes 6 steps or something like that. Eventually it'll get to an alpha keto acid, and then alpha keto acid either will be directly, usually it will be one of the metabolites that's directly in the citric acid cycle, like alpha ketoglutarate, oxaloacetate. So we are thinking about this removal of the nitrogen through the urea cycle and then the connection to the citric acid cycle that way. So we talked about some of these enzymes. We talked about transaminases in some detail, or aminotransferases. These are the PLP-dependent enzymes, and we did a little bit of mechanism on that. So we looked at the structure of PLP, pyridoxal phosphate, and then we noticed that it was interconvertible with pyridoxamine phosphate, and that whenever there's a transamination reaction, there's 2 substrates and 2 products that function in a ping-pong mechanism. So there's an alpha amino acid that reacts with the PLP cofactor in such a way as to transfer the amino group to the PLP cofactor to make pyridoxamine phosphate, and release the alpha keto acid as the first product. Right? So the first product, alpha keto acid. Then this transaminase will have pyridoxamine phosphate on it. That will be shown here. So we'll do the first. If we imagine that we're starting here on the enzyme, and the enzymes are named after the amino acids. So, for example, aspartate aminotransferase. Then the first thing that would happen is that this amino group would be transferred onto here to form pyridoxamine phosphate, and that would release the alpha keto acid. And then once we have pyridoxamine phosphate still stuck on the enzyme, then the alpha keto acid substrate binds and is converted to the amino acid, while this pyridoxamine is converted back to pyridoxal phosphate. Right? So this second substrate, so to speak, if this is the first one, this is the second one. This one doesn't bind until this product is released, so that's why it's ping pong. So that's the sense of the transaminase reactions. Transdeamination is a process that means a transaminase reaction. So these two, together with this glutamate dehydrogenase reaction, which is the reaction that takes ammonium out of the amino acid glutamate. All the amino acids here, alanine, other amino acids from the diet, are getting collected into glutamate, and then this glutamate dehydrogenase reaction releases ammonium from the alpha position to generate alpha ketoglutarate, and then this ammonium goes into the urea cycle. Okay. If we've got other tissues, especially muscle, glutamine is made in those tissues by the enzyme glutamine synthetase. And now here, this is a liver slide. So glutamine arrives at the liver, and this enzyme glutaminase converts it to glutamate, and then glutamate can spit out the nitrogen this way. Okay. So glutamate collects the amino groups, and then glutamate dehydrogenase removes ammonium. So even though the mammals, humans, or many vertebrates are generating urea as the excretion product, this enzyme still generates ammonium. It's just that it's not directly excreted. The ammonium is incorporated into another metabolite that shoots through the urea cycle, and it ends up being made into urea, so that then goes from the liver to the kidney to get excreted. Okay. Any questions about this part of it? Yes, sir.
So this is all current. Oh, it says right there. In the liver.
This is all liver stuff. So liver is the big metabolic organ, especially for things like waste removal, and nitrogen has to be gotten rid of. So
I have a question.
Yeah.
So will the incoming glutamine release two equivalents of ammonium, because when glutamine is
Yes. Yes. So this, you see that this glutamine is actually putting out an ammonium here, right, and then sending it to glutamate. So the glutamine does have released two equivalents of ammonium. You're right. Let's get to another slide, and then we'll modify that a bit. Okay. So transaminases. And we talked about the mechanism. So then this is the dehydrogenase reaction. This is an oxidative deamination reaction, by the way, because this carbon here in glutamate is oxidized. So CNH3H, the double bond node, that amounts to an oxidation. If we have an oxidation here, we can have a reduction of something else, and so that would be either NAD or NADP getting reduced. Okay. So this kind of offsets the energy requirements of the urea cycle a little bit, because in the process of nitrogen excretion, because here's the urea is generated, it's going to get incorporated into another metabolite for eventual excretion. That whole process of nitrogen excretion does involve the synthesis of reducing equivalent. Right? So that can, especially if it's NADP, because NADP, if it's NADH, then it can generate ATP in oxidative phosphorylation. If it's NADPH, it's used in synthetic reactions, like the NADPH that's made in pentose phosphate pathway. Okay, so again, transamination plus this oxidative deamination here is called transdeamination. Okay. And then we talked about this glucose and alanine cycle by which glycolysis in the muscle generates pyruvate, but there's an aminotransferase here generating alanine from pyruvate, and there's actually a level of blood alanine by which alanine comes back to the liver. And then the amino group from alanine can get put into the urea cycle, but the skeleton of it can be made into, that results from that aminotransferase is pyruvates, and then we can make glucose in the liver by gluconeogenesis, and the glucose can go back in glycolysis. Right? So it's like, this is really showing how the liver is making glucose so that the muscle can degrade it. In the process of this cycle, which is basically the liver supplying the muscle with fuel and taking back the sort of the fuel end product so that it will generate more fuel. it also is it's providing a route by which excess ammonia in the muscle proteins that are getting broken down can get sent over to the liver. Yeah.
Where does the glutamine that's coming from the muscle come from?
Where does it come from?
Yeah, like from the other side where glutamine comes in. From
It comes
muscle.
from the enzyme glutamine synthetase. Yeah, so it comes from glutamate. Right. So if you have so in muscle cells, if you're collecting nitrogen in muscle cells that needs to be excreted, and there's not really a functioning urea cycle to build up urea and send it out to the kidneys, which is what the liver does, then this process here can incorporate excess ammonium in the muscle to generate glutamine, and then the glutamine has to go out through the blood to get to the liver, and then this goes on in the liver, this glutaminase. So this is liver here, and this is muscle, for example. But it's also other tissues besides muscle. Okay. All right, so then we've got these reactions. So here's the extrahepatic tissue. So this is muscle and other tissues. Glutamine coming in. Glutamate also potentially coming in from the cytoplasm of the cell, or getting from alanine in muscle, getting generated through a transamination reaction in the cytosol, and then glutamate coming into the mitochondria. So what we have here is we have some reactions in the mitochondria. Most of the urea cycle back in the cytosol, the mitochondria is just this part, right? And here is the glutamate dehydrogenase to generate the ammonia. And the first step of it's not really the first step of the urea cycle, it's the incorporation of inorganic ammonia, costing 2 ATPs with this enzyme carbonyl phosphate synthase to make carbonyl phosphate.